זה קטע מסכים. כן. זה לפעמים אני לוקח רמז מאלוהי העיצוב, שיש בעיה. מה? יש מישהו?
סגור?
כן, 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 אני שם מיקרופון. רמקול. Hi there.
Hello.
Can you see? I'm trying to.
It's okay.
Okay. Yeah, I know you said come alone, but don't bring police. But I have a student here.
Nice to meet you.
Yeah. So thanks for the time. So I'm Professor Lewis, and this is Aaron, that's the PhD student, new one, and then you, Steven.
Steven.
Do we say the whole? My father's name is Steve.
Yeah, well, I listen to a lot of versions of my name, so you can call me Steve or Steven. It's all fine.
There you go. That's a CD that I'd like to listen to. So, how are you doing? Where are you now?
I'm in the Netherlands. We're doing pretty good over here. We still have a lot of the corona measurements in effect. So if we don't have to be at the lab, we have to work from home. So
Yeah.
so
it's like the research comes first. That we have that here as well. Yeah.
Yeah. We were locked down for about 3 months, but we could not go to the lab at all in the beginning, about a year back. And now we can go to the lab with about 50 to 75% of the normal occupation, which for us is fine. So you come in, you do your work, and then you go home again to write. So, but most of the days I'm in the lab, thank God.
Well, also you wrote a paper, so that's maybe a good thing.
Yeah. Yeah, we have multiple manuscripts running at the moment, so it's nice. But like, yeah, you do have to write together with your PhD students, etc. So we need to come together at a certain point. I think last year we had a bit of a delay in a lot of the projects, so now all of the projects are finishing. So we're like yesterday actually I submitted a manuscript about the serpent project that we were running. So
you should put me as
we'll be okay.
Requested reviewer if you want.
Sorry?
You can put me as requested reviewer if you wish.
Thank you very much.
Yeah,
Yeah, I will add you to the list.
Sure. I have a table of potential reviewers and a separate list for those who believe in alpha-1 antitrypsin. Yeah. You know more than I do. Some of them have this resistance for some reason. Like they're not open-minded.
Yeah, well, my problem with the serpent field is that this is a pretty old field. So
Yeah.
there are a lot of people in there that go like it should always be done like this, it's always like this, etc. And they can be very stringent about certain details. And you go like it doesn't really matter. They go like yes it does, and you go like okay fine, I will do it like that. But one thing that one of the lessons I got from one of my supervisors was like if the reviewer asks you to print it on pink paper, print it on pink paper. You just do what the reviewer asks you to do and just do it. But sometimes I think 2 years back we published something on antitrypsin mutants, and I had a really long discussion with one of the reviewers. He's like, it cannot be like this. So I proved him wrong with all kinds of data that's not in the paper. But at a certain point, I got a bit tired of the field going like, okay, it's all nice, but you guys got stuck. It's fine, but it's time for a bit of a new energy into the field.
I use that discussion area to try to educate a little bit, the scientific community. I'll invest like in the discussions. Discussion is usually not the place that they cut down issues, and it's important to educate them. There's a leap. I mean, I think it was more than 20 years with no release of anything new since the 80s.
Yeah, you can see that often going on in field. So I originally started in the contact system, so which is factor 12 and plasma kallikrein, those enzymes in the plasma. And also there, like people, they figured stuff out in the 60s, they figured stuff out in the 80s, and then somehow they froze and they were like cycling through the same stuff
Yeah.
again. And now suddenly you can see that they're changing their mind again. And now you can see they're actually going back towards what they found in the 60s, but they kind of ignored it in the 80s, going like, yeah, we kind of figured it out, but now we're going to go this route. And then for some way they got stuck. I think that's science for you.
Yeah, presently it's like science is taking a step back, being a little modest now.
Yeah, well, we have to, but I think sometimes there's also a lot of politics involved in the science, and like at a certain point certain people in the field, they get a lot of power and they will try to provoke their own thoughts, of course, or their own school, their own group, or however you want to call it. And then people really invest on that, and sometimes they're right and sometimes they're wrong, and then the
Yeah,
field gets stuck. And then if somebody new comes in that doesn't care about all those people in power and just does what they think is right, then sometimes you can see that field opening up again.
yeah, it's specifically Alpha One. I mean, I was in one. I'm in touch with the Alpha One Foundation,
Mm-hmm.
but I mean the actual those who actually discovered, worked on it, all the big names. They're old, but they're there. There was a meeting in Portugal a few
Mm-hmm.
years ago, and they were all there around the table, and it's like they know that it can't be that the Alpha One story is finished. It can't be that that's what we know. Just give it clinically to emphysema. They started to realize that it's more than that. Maybe then, like they had um, clinical trials started with a viral vector for Alpha One
Mm-hmm.
intramuscular, trying to give it intramuscular, like, almost like gene therapy, and then they measured the levels, and they presented those preliminary data that the levels were that it succeeded, but the levels were very low, so it kind of failed, really, really low,
Yeah.
like 1,000 times less. And they considered it a failure. And what do? More virus, more injections, more repeat. But like like in conferences, you hear the talking behind the scenes. They said that the patients actually benefited. They really
Mm-hmm.
had an effect, and it was so hard for them to cross that mental barrier that it's not about the amount necessarily. Maybe the purification of clinical alpha 1 does something that was never
Sure.
important enough to measure, and then we see things like they practically, how do you say, isolate alpha 1 with affinity to elastase. That's
Mm-hmm.
ridiculous. Like, that's the factory criteria is inhibit elastase.
Mm-hmm.
And that you and I know, Aaron maybe knows, it's a first year PhD, that's overrated, right? Like, so
Yeah.
anyway, so that older community now they're slightly divided, and there's one in Ireland. I don't know if you work with him, Gary McElvany.
No, I don't know.
It'll be interesting to connect us all. He's like different than the rest, and I mean they were all in the same time, getting the clinical alpha one into the arena in the 80s, and he's one of the
Mm-hmm.
few that now believes that as a serpine, as alpha one, there's more to the story, and they're looking more into variants and a lot more. All this in Ireland, uh, which for some reason has a lot of alpha one deficiency, genetic,
Yeah,
kind of disproportionate.
maybe
Like
because of the more family relations. Yeah.
yeah, there's like hundreds in a small area compared to, I mean, in Israel there's 50 patients, 50, in the
Oh,
entire country.
less.
Yeah, you have less. Well, it depends who the Vikings won over, and so yeah,
I worked on the hereditary angioedema. You can see the same, like in certain countries you see a lot, and other countries you can barely find them, and it has to do a lot with the heritage indeed.
yeah,
No, but I think also that the times have changed for antitrypsin now, that we can easily produce proteins, we can easily modify them, etc. That we can do more.
oh yeah. So okay, so
So the people that
hmm?
So yeah, I mean, the old generation are still kind of stuck in how they used to do it sometimes in their old days. And like now you can just basically you can make any protein that you want. It doesn't really matter. I think antitrypsin is a really easy protein in that case to modify and to produce and test even. So yeah, I mean, I
That's
think
a-
there's
what-
where the field needs to open up. That we can do more. In my group, we're currently really looking at changing antitrypsin specificity, but you can do so much more if you want to. But you need to just have a good thought. You need to have good systems to measure, and you need to be fast and then have a really good eye on what needs to happen for the clinical side.
So, are you a clinician? Are you an MD PhD practicing?
No, I'm a PhD. I do not have the patience to work with patients.
Same here, same thing.
But I work in a hospital, so I work here in an academic hospital here in Utrecht. So we can be pretty close to the patients if we want to. But
Nice.
in general,
Here with this
for the last 10
same,
years,
oops.
we find experts internationally that are close to the patients, and then we work with those people.
If you need with a deficiency or not necessarily.
Not necessarily. I mean, we're not a classical antitrypsin group, so we don't really work on the deficiency part yet. We have some ideas that we might elaborate in the future, but we're now currently for antitrypsin, we're mainly focused on, hey, can we change its specificity
Great.
towards other enzymes?
That's amazing.
So
We're like twin labs basically, but uh, no, seriously, we're also positioned in a hospital, and
Mhm.
clinicians come in and out, and so we have a lot of kind of ideas and samples, and and we're also trying to manipulate alpha ones, structure, sequence, all that to learn more about the structure. But what's interesting is we don't touch the specificity issue. We
Okay.
completely focus on the globular outside, kind of binding, allegedly
Backbone.
binding sites. Yeah, so that's nice because I mean, the first thing that we do is truncate it. Like it just for us, it's better if it doesn't inhibit elastase because we can see the other functions.
Hmm.
That's why I was so excited to see your paper because suddenly you can see the lenses that you see alpha 1 through that we see the same thing. Like it's time to strip it down and evaluate.
Yeah, I mean, you have those papers on conserpin, right? So they basically where they try to make one serpent backbone in which you can change these whole things. I think it's a nice idea. I think it was a very broad idea that, of course, in its application, you will always find problems with it. But I do think that, I mean, the serpents have a nice quality that they only target active enzymes, which I think is really great. I think it's their main advantage above, for example, antibodies. But their instability as a molecule is also their downside. So, for example, I'm currently for a different project trying to produce pi one that's a horribly unstable
Yeah,
serpent. And so
PAI.
if you can fix it, if you can make it smaller, if you can increase its longevity, or if you can make it more functional, then I think serpents can be a really powerful tool,
Yes.
especially because you're already so close to nature.
Yeah, that's a good advantage. I mean, it is. That actually, that's one of the reasons that when we test our things with like a clinical horizon closer, then we first of all go for the topical treatment, like outside, like on the skin in a wound or a scratch or surgical suture, because at least then nobody will say, well, you're injecting something foreign,
Mm.
if we mutate it. Even trying to correct tissues in an ear damage, like inner ear damage, and eye, like the epithelial lining of the eye, if there's a scratch. These are the areas we run with testing some of the fake variants that we create.
Mm.
Because if it's friendly, if it works and speeds up the healing, then at least we have a half a door open for outside treatment, like non systemic.
Mm.
Yeah.
I think with modifying proteins, people are always very afraid of that something might happen, of course. But honestly, if you look at the antibody development world, I mean, you're constantly doing it there. You're changing loops, and then you're humanizing the antibodies, etc., etc. And there it has been accepted. And of course, you need to test it. I mean, for sure. For
Always.
example, I'm now currently involved in a company. We're trying to make a new protein for thrombolysis. And all the venture capitalists go like, yeah, what about immunogenicity? Yeah, okay, true. It might be there. We have to test it. But if you do not try, if you do not see, hey, can we alter its function? If you do not even show it in an animal model and show, hey, there's value in targeting this or not targeting that or changing something, that's the first step, showing that there's value. And then the second step is getting there in a way that it doesn't give you major side effects, of course.
Yeah,
But
I
if we
think.
don't try it and then don't go for it.
Yeah, I think they suffer from the regulatory authority, those companies. I mean, they'd love to have a product, but I think it's getting also stricter on them. Uh,
And
so,
for example, I mean, you can see now with the vaccination for COVID-19, right? I mean, with the AstraZeneca stuff, like the side effects is percentage-wise is very minimally, but people make a huge fuss about it, and it's so much better than having COVID yourself.
yeah,
But yeah, people go like, no, everything should be perfect, and honestly, it's impossible to make a drug that's perfect. And when we're
except for
changing
Alpha
the
1.
rules, which is fine.
Except for Alpha 1.
Yeah, that one's perfect.
Well, you know what? I give lectures sometimes on our data with clinicians, and suddenly they ask about side effects, and I say, but there's no side effects for giving Alpha 1 for people who are eligible, or, you know,
Mhm.
and they just refuse to get it, to accept it. There's no side effects. And we had a big clinical trial in Israel with diabetes type 1. All the
Mhm.
work on Alpha 1 with diabetes, it's kind of almost like my fault in the postdoc fellowship. I asked the question and it started to explode. And we had a double blind study in Israel with kids. And so it was really double blind. I mean, everybody was completely blinded and multi
Mhm.
center. And in my hospital, I started to ask the staff if they see anything. And they said, well, we thought we were hesitant to tell you, Louis, but one of the patients had anaphylactic shock. So I said, was it with Alpha 1 or not? And they said, we can't tell you, we cannot tell you. And when it ended, it was the albumin group.
Yeah.
Completely, right?
Uh,
Maybe if they gave him Alpha 1 in addition, he would actually wake up and stay. Yeah, it's. So, um, I guess, yeah, I think we're just connecting. I mean, because, it makes sense to combine forces. And if you wish, I can kind of give you a quick overview of what we're doing. If that,
yeah.
okay.
I can show you some of the data that we just submitted to show you
Okay.
basically which direction we have been moving on. It's
Okay.
confidential, of course, but I can show you via Zoom. That's no problem.
It's okay. I have amnesia from the side effects of the drugs, so of the vaccination.
That's a positive thing for your PhD students, right? Like I can tell them everything. Tomorrow he has forgotten all about it.
Yeah, but you say free food and they drop everything. Never mind. We've been there. Don't worry. We're not accusing. So I'll give you a peek, like just in general for the outline. In most of the years since I finished my postdoc, so that was 2006, most of the
Mhm.
time it was showing what Alpha 1 can do using the clinical
Yeah.
grade in many, many, many models. And at some point it was a bit embarrassing how many things it can achieve in different models. And
Mhm.
I always knew that I'm going to have to kind of switch gears at some point and go more for the structure, mechanism, not just outcomes. And maybe, I guess, around 10 years ago, I started to work more on the structure in addition to everything. We had a
Mhm.
plasmid from Stanford. Stanford Genetics had a gene therapy project, and they just had this huge plasmid with human alpha 1, including introns and exons. Very impressive. Like this plasmid is 28 base pairs, 28,000 base pairs. It's huge.
Mm.
Yeah, it's like the
Yeah.
vector is a small area. And that was their success. All they needed is to put it in cells, and express, and we took it and we're starting to use it now to modify areas. And the whole concept is that we're trying to move away from the proteases just so that we can gather information about the binding qualities, because there's things that it does without inhibiting proteases. And then there's other models where it looks like it's very much protease dependent. And so there we started two strategies. One, just a screening of potential sites on the surface of alpha-1, globular structure, so outside the RCL. Screening based on, how do you call it, like evolution? Evolution. How do you say evolution? Evolutionary, sequences of alpha-1, primitive ones, where there's some flexibility on the surface of alpha-1 that will at least allow the protein to be expressed. So we based it back to consensus, it's called. So we had this map of 50-something sites on the surface, and we started to mutate them and bombard and collect ideas of some candidate areas, and we did find some binding proteins which are interesting and areas of alpha-1 that bind things. It's basically a very, it does bind outside the RCL very nicely, several things, especially lipids, by the way. I don't know if you're like me, but I'm afraid of lipids, but I mean, scientifically I'm afraid. What?
I work in a coagulation lab. We love lipids. We need lipids.
So yeah, you should look at this. When you use alpha-1, do you get clinical grade alpha-1? Like from which company? Zimera? Do you get?
No, we all antitrypsin that I work with is produced in-house.
Okay, so it was really ridiculous, but one of the companies makes alpha-1 that's so purified that it works less good.
Yeah.
And the other one, two other companies make it, and there's a little bit kind of, you can see the residuals in the sample, in the outcome, and they work better. And it turns out that it needs palmitate in order to work. It needs lipids. And in the blood, basically it's a small lipid carrier, just like albumin, but it has some capacity. And the ridiculous thing was they took the company's Zimera, the one that's too pure. It's like a success in the protein world, and you mix it with palmitate and it works fine. So it's like the environment has to replicate the plasma.
I can imagine. So is that necessary to stabilize the SERPIN function, like the stability, or is it just like bringing the lipids along has a secondary function?
Well, I know that it was all in vitro. I mean, we're talking about cell assays. This was never. Well, I mean, the things that they found is if you inject it in vivo, it's fine, because then it goes into the system.
Because
but you try to replicate.
So it binds the lipids from the plasma.
Exactly, and you try to replicate it in a plate, and there's suddenly differences. So it was all in vitro, but a nice outcome.
Nice.
Almost like, the serum replaces many of the things that we manipulate.
Yeah.
So then we did the screening. We found a few candidates. We have areas on alpha-1 on the outside that are really interesting, and we narrowed it down. In parallel, we started to look for areas on the outside and mutated a few of those. And Aaron, Aharon's, how do you?
Aaron.
Even in Hebrew it's hard. Aharon, like a aha moment, Ron. Aharon.
I'm gonna mess up your name a few times. I'm really sorry, by the way.
No, it's okay.
That's nice. That's good. You get a V. So so his project is very ambitious. It's something that I started to kind of think about a few years back, but looking at the structure of alpha-1 on the outside and going way back to the Salamander, because they can regenerate a limb, kind of. And there was another group that published something. That's where suddenly I got like this confidence. They found an elastase inhibitor in the Salamander that's required for regeneration of a limb, but it's not alpha-1. But if you look at the sequences, there's elements of alpha-1 there, kind of hidden.
Is it also similar to alpha-1 antitrypsin 2 that we have as humans?
I didn't check, but it's way inside kind of the conserved area of alpha one backbone. I
Hmm.
mean, it may mean nothing, but you cannot. It doesn't really immediately look like alpha one. But a lot of the sequences are very much preserved, and so it caught my eye. And now we're thinking that if we take everything we learned about the surface of alpha one sequence on the outside, and see how we can navigate towards tissue regeneration using some form of alpha one, that'll be amazing. That's his project, and so
Cool.
it involves, right, a little ambitious but very very nice images of salamanders. Uh, yeah, because we see with alpha one, native alpha one, we see that it helps blood vessels form, and that it's really pro healing, and so that's his project. Um,
That's really cool.
we have some work on diabetes, like what happens when alpha one becomes over glycated, because the whole story started with diabetes and islets. We have some work on GVHD on bone marrow transplants using the clinical grade in clinical trials. Yeah, that's where we are, roughly.
Nice. Nice. All right. Cool. Uh, I will share my screen with you.
Nice.
This has been a presentation that I gave to to a group of people involved in one of my grants. So it's a bit on the nose for you guys probably, but
Okay. Oh, I had to give a presentation to Chinese people.
yeah.
I just put up a picture of a Swiss Army knife. That's it, and started to talk. There was no.
So yeah, I so I originally started on working on coagulation enzymes, and those coagulation enzymes contain cleavage sites, and we try to predict which enzymes cleave in this cleavage site. So that's how we got to serpents in the end. So our focus of our group is more or less to modify serpents in their function by changing their cleavage site. Now, to predict which sequences are interesting for certain enzymes, we found that to be pretty tricky, as basically the entire serpent community has found it to be tricky. So we tried to find a way in which we could do it better. So what we decided to do is that we took the 8 amino acids surrounding the cleavage site in the RCL, and we started to mutagenize them with all the natural amino acids at each position. So we have 8 libraries made based upon antitrypsin Pittsburgh, and we changed each position, reproduced them, and then we tested them against several enzymes to see how that would affect our behavior. And this is one of the heat maps. So we, as I said, I work in a coagulation lab, so we mainly focus on coagulation.
Yeah.
And you can see now the effect of amino acids mutations within the RCL on their inhibition. So you could see, for example, for thrombin, it really requires arginine at the P1 position, but you can see diverse behavior than per enzyme like this.
So,
And if
so
you
the
would combine
brighter,
this data,
yeah, the more
yes,
it's bright, the brighter the
yeah,
better
the more yellow it is, the more inhibition you see.
inhibition. So the serpent is better. Yeah.
Yeah, the RCL sequence is better
Better.
for inhibition. Yeah.
Nice.
And you can see that well, for the P1 position, usually it's an arginine. Some, for example, factor 11A and thrombin also prefer a lysine, but that's mainly determined by the protease itself. But then for the rest, you can really see that the behavior between the enzymes is quite different on the other positions. So it allows you to generate specificity profiles, basically.
Nice.
So we took this data and we tried to build a prediction model for this, and we compared it to libraries developed based upon peptides. So you have those peptides with 3 amino acids and a chemical group, those substrate libraries. So we compared their prediction versus our prediction. For example, this is activated protein C. So this is basically if you would take those peptide libraries and see how it predicts. If you say, okay, you predict the inhibition from 0 to 100%, you're on the x-axis, you measure the actual inhibition, you can see there's no correlation. So the prediction is really bad, but via our system, we got pretty much there for activated protein C. So we can now really start designing serpent specificity for what we wanted. So for factor 12, thrombin, and activated protein C, we got pretty good correlations if you look at the black ones, while the peptide libraries were all over the place. For factor 11A and plasmin, we got lesser results, but still workable. And then for plasma kallikrein and factor 10A, we got pretty bad results actually, but at all times, it was always better than the peptide library that we used again. So it's not a perfect approach for many reasons, but the alternative is to build 26 billion combinations of serpents, which is just not doable.
Can I ask a technical question?
Yeah.
So these are all enzymatic assays, right?
Yes.
Are they? How specific are they? We're struggling with a few. So you put a substrate in and the color comes out, but are you worried about overlap, like with another enzyme?
These are all purified, so we actually know which
Right.
enzyme is there in only. So this is quite specific, yeah.
Yeah. So artificially place them together, not like a serum samples capacity.
No. This is completely purified, just to check. Yeah. So this is specific.
Great.
So to see if this platform would hold any value, we decided to develop a new serpent. So we took this paper from the Huntington lab. It was published in 2017, where they took alpha-1 antitrypsin in Pittsburgh and they redeveloped it to inhibit activated protein C as a treatment for hemophilia. And they actually have two papers on this. So this is the blood paper, which they did the original thing, and then they used random mutagenesis later on to try to improve it, but they failed to improve the efficacy or the specificity further. So basically, they said this is the best that we can do, at least in what they published. So we took the optimal sequence from our library to inhibit APC. So this is KNPR-SIPA, but it's not specific enough. So you can see it inhibits APC pretty well according to our prediction model, but still a lot of thrombin, factor 10, etc., etc. So we started changing the RCL further, and we got 3 sequences in which we have more specificity than we wanted, or the better specificity as we wanted, and we produced those. Now you can see if you look at activated protein C inhibitor, this is the wild type. It hardly does anything. The KRK published by Huntington gives us about 30% inhibition. And you can see that the variants that we built are stronger inhibitors. So you get better APC inhibition. So as a treatment, this should become more potent.
Nice.
For thrombin, we didn't find any inhibition, which is good because you do not want to inhibit thrombin. And for factor 10A, you would find the same thing.
Wow.
For factor 11, we found that the published variant by Huntington gave us still some inhibition, while our variants did not. So this is an increase in specificity that we found. And we found a similar story back for plasmin for one of our variants, while the other 2 are about equal to the KRK variant. Now, if you want to inhibit plasmin or not in the hemophilia patient, there's a very long discussion that you can have, but the goal of our story was to make a serpent that was more specific or more potent.
Yeah.
The only thing that was really weird was the factor 12 inhibition, which is not really relevant in these patients, but we saw a slight increase in that. But for plasma kallikrein, for example, we also had an increase in specificity again. So we were quite happy. And so the prediction platform is not 100% accurate, but it does give us a pretty good platform to start building serpents. So then we took a look at plasma, of course, because these were all still purified experiments. Now, if we do coagulation experiments in normal healthy donor plasma, nothing happens. So you don't see any off-target effects, while Pittsburgh prevents coagulation in a concentration-dependent manner. And we see the similar story in factor 8 deficient patients, so that's hemophilia type A, or factor 9, which is hemophilia type B. So we don't see any effects, and that's because APC is not activated under these conditions. You can see we show the same in clotting assays, and we actually saw that the KRK still has a weird effect in the clotting assay. They also described it in the paper, but they say that it's normal. But if we do it at specific concentrations under certain conditions, we find that it does have an effect on coagulation, while our two of our three variants that we built do not. So it's an increase in specificity again. Now back to the thrombin generation. Now, if we add thrombomodulin to the coagulation experiment, you activate APC. Your coagulation potential from 100% goes down to say 50. And if you add the serpents, then you start inhibiting this APC, and you can build it back up to almost 100%. And you can see that the variants that we created in blue, green, and purple are stronger than the ones that was published by the group of Huntington. Now, we did this with increasing concentrations, of course, to show that we didn't pick the concentration that only worked nicely, but you can see that at all concentrations, improved APC inhibition is beneficial. Now, in the factor VIII deficient plasma, so that would be a plasma of patients, for example, you can see that it's the same story. It works better than what they published. And in the factor IX deficient plasma, we found the same thing. So that was really great. So finally, we went to a factor VIII deficient mice. We collaborated with a group in Paris, in France. Now these mice, they bleed. But if you give them back factor VIII, you can see you can restore the bleeding, so they hardly bleed. But if you give them alpha-1 antitrypsin wild type, which doesn't do anything on this pathway, you can see that these mice bleed around 800 microliters in 30 minutes. Now, if you inject the KRK, the variant from Huntington lab, you can see at 7.5 milligrams per kilo, nothing really happens. But at 15 milligrams per kilo, you can see that the blood loss is recovered by about 50%. So that was really nice. And you can see that the variant that we built in green can already do this at 7.5 milligrams per kilo. So you can just see that you generate a bigger therapeutic window because you have a higher efficacy and more specificity.
Yeah, and also it looks like the group is a bit more, how do you say, condensed, not so scattered,
yeah.
Maybe.
Yeah, so basically the interesting thing is that if you go from 7.5 to 15, it doesn't really matter that much anymore. So somehow this model is evening out around about 50% of the blood loss recovery.
Exactly.
This is a bit weird. We cannot completely explain it, but it seems that APC only contributes so much to this blood loss story, and that this is the maximum that you could do. It has been described also for other medication, but it shows you basically that we can design serpents that are more powerful and therefore work at lower concentrations. So you improve the therapeutic window. We made it more specific, so you get less off-target effects. But of course, we're limited by the system that it is. So yeah, that was basically this what we just submitted, and this is also what the group is mainly working on at the moment. Basically, we have a main focus on trying to change serpent specificity and to really design it. But we do have plans to start, for example, improving backbone stability, et cetera, just to make serpents a better therapeutic agent in general.
Yeah, well, especially if you find a nice variant that you make with RCL, you'd like to then manipulate the rest.
For example, or to target it somewhere specific. So, for example, if you look at antitrypsin wild type, the reason why the concentrations need to be so high, of course, is because it's in the wrong location. So can we improve it? Can we shuttle it to different things? And I think that's always necessary if we ever want to make gene therapy for alpha-1 antitrypsin interesting.
Yeah.
But for example, in 2017 we did some experiments in mice, and we showed that in an inflammation model with one of the serpent variants that we built, we already had efficacy around 10 micrograms per milliliter, which has already been achieved in humans with gene therapy. So it means that if you make the efficacy high enough and get it into the right location, then gene therapy for these chronic diseases is something that's really interesting.
Yeah, and I'm also thinking, in addition, that the purification process of the clinical stuff is pretty violent.
Oh yeah.
That's what we spoke about. So it's probably missing some things, but then inhibiting elastase. But gene therapy has the product immediately released into the system without any cleaning. Yeah, we have these mice that are that they have the human gene alpha-1 human gene in the lungs.
Hmm.
It was developed in Canada for smoking studies, and they almost threw away that colony. But I just needed a mouse that's born with alpha-1, so I can inject human alpha-1. And you know, maybe you have this thing as well, but it's kind of alpha-1 always has like a surprise around the corner somewhere. Like you think you build this theory and it's the opposite. And so you can barely measure it, like it's barely detectable because it's only lung produced into
Hmm.
into the immediate lung area, but then a little systemic. But these animals display things, the homozygote ones display
Hmm.
outcomes that are sometimes even better than injecting or whatever. And those levels are really hard to explain. When you talk about discussions, almost every paper 10 years ago that we had with an outcome with these mice, we couldn't publish
Hmm.
because nobody believed that such a small level could do anything. And we keep finding these outcomes. Yeah, so maybe it goes with like the way that the native one presents itself in the blood without all kinds of interactions before.
Yeah, that could very well be. I mean, you also see this, for example, with C1 inhibitor esterase, the other serpent G1.
Yeah.
You can see that the recombinant variant, if you inject it, disappears really quickly because it's wrongly glycosylated,
Oh
but
yeah.
the effects remain very long. So it seems that the distribution of that serpent variant is different than its native variant, but probably gets to spots where it does the work better at that point. So while it's an imperfect molecule for certain aspects, it's still, you can still really treat patients with it. The
Yeah.
problem with that molecule is if you want to go for in vivo expression, because it's so bulky and so glycosylated, it would be better to, for example, modify antitrypsin to do the work for instead of it, because
Yeah.
it's just more easily expressed. But I think a continuous expression on even low levels, maybe not even perfectly, but lower levels will already benefit patients so much more than the continuous injection where you have those peak levels in which things are really effective and then it goes too low again, or what remains is not functional enough or functioning in a different way than we think it should be, that you're missing out on a lot of things. And that's the reason as an effect, the disease progresses more than you think it does, even despite this treatment.
Exactly. That's what we mentioned in the beginning. Actually, it's nice that it comes around. Like the Alpha-1 Foundation, slowly the scientists start to realize that the low levels that are produced inside the cell, of, like a viral infused person, are just as good or better than the. It's but they have to let go of the success of infusing Alpha-1
Yeah.
once a week, twice a week, and I remember I tried to calculate like the area under the surface of the small levels that we have in the mice, but it's constitutive. It's
Yeah.
completely unregulated small levels, and it turned out to be almost as high, if not higher, than the normal distribution, which peaks and then disappears. Almost like if you keep, if the faucet is dripping, you basically water more than
Hmm.
opening it for a few hours and closing.
No,
Uh,
for sure.
and also, we found a hint of something that may be also relevant to you in a nice way. The spike that is followed by the disappearance could be the binding to the membranes of monocytes in the blood.
Mm.
So it's positioned on the membrane, and there's work mostly by Sabina. You probably follow her papers. Sabina from Germany. Jungst something. Family name is like a signaling cascade. Uh,
Hmm.
but yeah, she's also working on this a lot on the clinical side on alpha-1 alone. And anyway, it turns out that it might be working on this. It binds to the surface, mostly to cholesterol residues on the surface of cells. And so when it drips in, when it comes in at small levels, it kind of saturates those cells. So
Yeah.
they have more on it. And we literally found that if you take monocytes from a mouse who's homozygote to the gene away
Hmm.
from the mouse and you test them in a plate, even though they're not the expressing cells, they don't express more alpha-1, And even though
Mhm.
you wash it out, they already behave in a more favorable way than the equivalent wild-type mouse. Maybe
Yeah,
because
I can especially think about like with monocytes and those type of cells. That's really the long-term exposure to certain things or the long-term treatment, even if it's at low levels, can have dramatic effects on those type of cells.
yeah. Luckily for the better. I mean, usually it's a good thing. Like in the bone
Mm.
marrow transplant setup, we actually were able to get alpha one into the guidelines. So when a bone marrow transplant turns into GVHD and steroids fail, alpha one is
Hmm.
already in the guidelines to give, and that's part of the story and the science behind it. We found that even the donor of the bone marrow cells, if the donor had higher alpha one, the outcome of the transplant was better. It's kind of amazing how it's kind of under our nose all this time. Uh,
Yeah, you can see this also with different serpents. I think C1 inhibitor is also being now tested on bone marrow transplants, indeed, for a similar function.
Makes sense.
To yeah, so I mean, there's a wide world in which you can do the application. We're currently staying outside the native antitrypsin wild type functions for now, because we came from a different world basically.
Yeah.
We came basically in via Pittsburgh, so that was the focus
Flew
so far.
from Pittsburgh.
But yeah, to and then it's the easy part, of course, to redesign RCLs. But I think I think maybe one or two more papers will come out in that aspect, and then we'll probably start moving towards the backbone more
So
to
yeah,
see what we can optimize.
we can brainstorm when you get to that stage. It will be interesting to revisit and see what we have. It could be a very nice collaboration if you wish in that area. It's like an endless world over there. Really, it's full
Yeah, and I
of
think there's still a lot to do, right, for the patients, and I like those challenges.
yeah, completely. And it's funny because we're staying away from the RCL because we're afraid of the coagulation system. So it's like, I keep telling my students, whatever you do, don't touch the RCL, because who knows now it's going to cause a bleeding disorder.
Why you need to mess around with it so much.
Oh yeah. No, but I mean, because we're going for the clinical future of it, so we just don't want to have even a scarce hint of accidentally interfering with the cascade.
Yeah, I mean the same goes for you guys, right? So if you want to measure stuff towards coagulation or that type of experiments, then either send us some of the protein or send your PhD student with some of the protein, and they are more than happy to stay over. Well, if the situation becomes a bit better, for a few weeks to do their coagulation experiments. If you're afraid of those type of things, for sure.
We're afraid. We did
Yeah.
thrombin one evening because we had an angiogenesis paper, and we saw that there's more blood vessels forming based on
Mhm.
more bleeding when you pull out a transplant. And in the literature, you see thrombin in the same story. You have to exclude it.
Mhm.
And the student, she was almost. She thought that's it. We're inhibiting thrombin. We're probably doing more damage. And I told her, you know what? Give me your plasma, and we took some thrombin from the freezer. With
Mhm.
without alpha one, with it, and super concentrations, and then allow it to coagulate and flip the tubes. And it was like day and night. You see that it doesn't interfere with thrombin, but you put
Oh, I'm thinking about the experiments that one of my students recently did. I mean, you can add so much alpha-1 antitrypsin wild type to thrombin, nothing will happen.
yes, but we had to show it. I mean,
Yeah, for sure.
it felt ridiculous, but it's in her posters. But for the paper, we really didn't need to show like upside down tubes. But it was really like a nice thing to see in the evening. It was very dramatic, scary. If that one falls as well, yeah. I mean, if the serum falls down, that's it for us. It's like,
Yeah, yeah, you can see the entire project go
yeah,
like
yeah,
that.
that was nice. So, so you said that this paper was just submitted.
Yesterday.
Okay, I'm keeping my fingers crossed, and if you stumble into any problems, then I'd love to help. Uh,
Thank you very much.
yeah, and You know what? There's another small question. That how do you, when you purify alpha one, what's the basis? His tag, or
We put a strap tag on there, so it's at the end terminal site. The nice thing about the strap tag is that you can almost do a one-step purification.
That's great, because with his tag we're having kind of some good ones and some difficult ones, and so that's nice to know.
So
We'll
it's
try.
STREP strap tag. It's from a company. It's I think it's 8 amino acids, but we put 2 on there after each other. So it's a buy tag, and you can just buy the column material. And the only thing that we do is we produce in hex cells.
Yeah.
And we take the supernatants because the protein is secreted.
Right.
And we concentrate it and we change the buffer. So it goes up in pH to around 8. And then we apply it to the column. And via this way, it's almost a one-step purification. So for the mouse experiments, we do gel filtration afterwards. But we hardly see any contaminants coming in it. So it's almost like already, I think it's 95% pure already.
That's great. And when you go across variants, it's not like they start to behave differently too much. It's like
for purification, no, not at all.
Okay, nice to know. Great. Yeah, well, we'll try to apply some of that. Okay, and yeah, basically, I'm just thinking wider. Like, if you want to test things in vivo that we have running already, all these systems that I mentioned are running. Like scratch eye or like ear damage in mice. All that is running, and what I'm doing is switching around with the recombinants, collecting evidence. If you feel like any one of these models is relevant, and it's small amounts because we're sticking to the local application. So it's in the ear, it's on the eye, it could be on a scratch, like on a surgical suture. It could be very local. So if you wish to kind of do a proof of concept of one of them, you can also.
We're building a new serpent that specifically inhibits bradykinin production. So for the inflammatory pathway. We're currently testing it. It looks pretty good, but we might actually drop by with that because inhibition of factor 12 and PK has shown to have effects on tissue damage regeneration. So we might see if we can book a similar effect on what they saw in the knockout mice.
Great.
For sure.
Yeah, because that probably goes to the conserved area that's doing it, not so much the. Okay.
It'll be really cool.
Yeah. So you're welcome over, and then if we stop by in India again.
Utrecht.
No, I'm kidding. We'll start in Amsterdam, obviously, for obvious reasons, and then.
Good, and then just work your way through towards Utrecht. No, that's excellent.
Yes.
I think it's half hour in a train.
Everything is half an hour in a train in Holland.
Some things are 45 minutes. Don't underestimate us.
Okay, no, that's good. I have a colleague in Utrecht, but he's a clinician who works on fungi with alpha 1.
Okay.
What's his name? Mihai Neta, something like that. Mihai Neta.
I don't know.
He's deep into the
send him if you can send his name in the email towards me. We'll look him up. Maybe we can also have a collaboration.
Sure, I'll send it to you. It's
cool.
You better read about fungi before, because it's a bit embarrassing. Like, he knows everything about fungus and infections.
Excellent.
Okay then, so we'll keep in touch.
Yeah, for sure. It was nice meeting you guys.
You too.
Thank you.
Let's hope for a conference somewhere. That'll be great.
For sure. I'm ready for one again.
Yeah. Okay.
Hey, have a good morning, guys.
Bye.